Adriana Padilla,
Yes.
Joe Panetta,
Here.
Francisco Prieto, Robert Quint, Al Rowlett,
Here.
Jeff Sheehy,
Here.
Oz Stewart, Jonathan Thomas,
Here.
Art Torres,
Here.
Diane Winokur. You're on mute, Diane, but I see you, so I'm going to mark you as present. Okay, JT, we have a quorum.
Thank you, Maria. This is the latest in a series of meetings we've been having regarding COVID-related projects recommended for funding by the GWG, which this week met on Tuesday morning. Proceeding to the action items as before, first, before we get to the actual projects, we need to vote on the following: consideration of existence of emergency situation. Emergency situation means any of the following as determined by a majority of the members of the Independent Citizens Oversight Committee. A, work stoppage or other activity that severely impairs public health or safety, or both. B, crippling disaster that severely impairs public health or safety, or both. Do I hear a motion to approve?
Yes.
So moved.
This is David. Yes.
Moved by David Higgins, seconded, I believe, by Dave. Was that you?
Yes.
Dr. Martin.
Thank you.
Any discussions by members of the board? Any discussion by members of the public? Hearing none, Maria, will you please call the roll?
Anne Marie Dulege.
Yes. Yes.
Thank you. Isabel Duran,
Yes.
David Higgins,
Yes.
Steve Julesgard,
Yes.
Dave Martin,
Yes.
Lauren Miller, Adriana Padilla,
Yes.
Joe Panetta,
Yes.
Francisco Prieto. I know you're on, oh, you're on mute, but I see you. Thank you. Robert Quint, Al Rowlett,
Yes.
Jeff Sheehy,
Yes.
Oz Stewart, Jonathan Thomas,
Yes.
Art Torres,
Aye.
Diane Winokur. Thank you. Motion carries.
Thank you, Maria. On to item 4, which is consideration of applications submitted in response to special call for COVID-19 projects. I will turn the meeting at this point over to Mr. Sheehy.
Thank you, Chairman Thomas. Dr. Sobrano, do you have a presentation for this round?
Yes, I do. Okay, thank you, Mr. Sheehy. Doug, do you have the presentation to put up, or would you like me to do that? Okay, thank you. You can just skip over to slide 3. So good afternoon, everyone. So we by now are pretty familiar with the COVID-19 program. We've obviously been hearing quite a bit about it and have just had a couple of board meetings on it, so I won't belabor this. I do want to point out that for this particular cycle, all the applications that came in and that were reviewed were all in the discovery arena. So they were all DIS2 applications. And so a reminder that the award amount for these types of applications is $150,000 with a duration of up to 12 months. Next slide, please. And these are the kinds of projects that are intended to identify a candidate for development, either a therapeutic or a tool of some kind. And we set ambitious 6-month goal for all these projects. And in this case, for all the DIS2s, to have data for a viable candidate that could progress quickly to the clinic. Next slide, please. So for this cycle, there were 13 applications that were reviewed. There were 3 that were scored in the fundable range and thus recommended for funding. So the total applicant requests would be $449,000 400,000, well, 450,000 basically. There are still 2.9 million available in the COVID-19 program fund. Next slide, please. And so the three applications that are recommended are shown on this table. The first one is 11947. And so this is an application that had the score of 95, and it's entitled Persistent Off-the-Shelf MeAce2 CAR IL-15 NK cells derived from CD34 positive cord blood stem cells to prevent and treat COVID-19. And so what this application proposes to do is develop a cell therapy of natural killer cells that express ACE2 receptor. That's the receptor that the COVID-19 virus SARS-CoV-2 attaches to, as well as IL-15, which helps those NK cells persist. And so they've engineered this construct to use as a cell therapy and intend to develop that for use in patients with COVID-19. The second application is 11920. That one received a score of 90, and it's entitled Application of Prostaglandin E2 and BPV, which is a muscle stem cell targeting therapeutic. It's a combination drug to treat COVID-19 associated diaphragm atrophy. And so this is for patients suffering from ventilator-induced diaphragmatic dysfunction, or VIDD. And so this occurs in patients who develop a complication from having long-term mechanical ventilation, which happens to be true for many COVID-19 patients. And so what this therapeutic intends to do is to induce and treat patients and treat the muscle atrophy in the diaphragm of these patients. And then the last recommended application is 11941 that got a score of 86. This application is entitled Identifying HLA Class 1 Restricted Peptides that Induce CD8 Positive T Cells Against SARS-CoV-2. And so the goal of this project is to identify peptides from the SARS-CoV-2 virus that can be utilized and developed as a vaccine for the treatment of COVID-19. And specifically, they are developing peptides in a different and novel way that can induce most specifically CD8 positive T cell responses, which are believed to be the most long lasting and probably the likeliest mechanism to allow recovery from COVID-19. So those are the recommended applications. And Doug, you can put on the screen the spreadsheet that shows the remainder of the applications along with these three. But that is the conclusion of my presentation, Mr. Sheehy.
Thank you, Dr. Sembrano.
Excuse me, this is Oz. Can you folks hear me?
Yes.
Yes. Thanks, Oz.
Thank you.
Thank you, Dr. Sembrano. So at this point, I will take a motion to move any projects in Tier 2, the unfundable range, into Tier 1, the fundable range, if there is such a motion. Is there a motion to do that? Hearing none, I will then take motions on Tier 1. I'm going to step forward with a little bit of chair privilege here because I did want to separate out application, I'm trying to find the number, 11941, because I would like to move that forward for funding, but I would like to move that forward with funding with an amendment. And the amendment would be a condition that the applicant take, and I believe the applicant is on this call today, but that the applicant add work or shift work in order to take into account HLA types from populations that are disproportionately impacted by COVID. In the review, the reviewers noted that the HLA types that were going to be used for this vaccine were from Western Europe. And as we all are way too painfully aware, this COVID pandemic is disproportionately affecting certain communities in our country. And I will note that the reviewers expressed the sense that APCs, which I take to mean antigen presenting cells, can be generated for these other HLA types fairly quickly in as short a time as potentially a week. But I think if you look at your comments from the review in the summary, in two separate places, they note the lack of HLA types from disproportionately impacted populations. So with that condition, I would like to see if there is a second to a motion to approve that application. And then we can deal with the other two after that, if the rest of the board will indulge me for that.
Mr. Sheehy?
I second.
Yes?
Bell
Can you,
seconds.
can you please repeat the application?
Second by Ms. Durand. Pardon me?
Can you please let me know which application number this is again? I'm sorry, I didn't catch it.
11941. It's the
Okay.
vaccine application.
Yep, thank you.
And who was the maker of that motion, please?
Well, I did Beth, Jeff Sheehy.
Oh, okay. I'm sorry, Jeff. I missed that. Thanks.
Well, that's okay. Is there any board discussion on this motion?
I'm sorry, the research is related to stem cells, right?
Pardon me?
The vaccine issue is related to stem cell.
I suppose. I mean, they did make the comment in review that AIM 3, which is the weakest aim, was thrown in to make it relevant in stem cells.
Okay,
I do
just
think
wanna
this
make
is
sure we're
important
consistent I just want to make sure we're consistent with the board policy we adopted recently.
Yeah, I also note that reviewers felt that this was a very important project because there absolutely is a need, according to reviewers, for vaccines of current, for backstop vaccines if current projects don't succeed. And this is an incredibly novel approach and kind of what we were trying to get at last week. But in any event, is there additional board comment?
Well, but at the end of the day, if we approve this project and it turns out that they did not use stem cell research, will that violate what the board's action took last week?
The board failed to take any action last week, Senator Torres. And as I stated, AIM 3 does indeed use stem cells and is part of the project.
Well, I beg to differ. We accepted a motion to make sure that any vaccine research would be related to stem cells. Is that correct, Gilbert?
I thought that we did not accept a motion to do a vital research opportunity. I don't think we did we then changed anything other than what we've currently been doing.
So this application is eligible and fits within our current eligibility criteria. It includes the use of stem cells, and so therefore it is one we could fund.
Right, which we affirmed last week. Okay, thank you.
Isabel, may I make a comment?
Please.
I appreciate that you brought this up because in my read, I was trying to look for some recommendation, not necessarily understanding totally where such a recommendation for proportional and equitable research is required, where we have to make an amendment, where we need to bring the researcher in for the talk. So I appreciate that you recognized this, and I would hope that somehow we don't have to keep doing this, but that this could be a very inclusive, either an amendment or one of the requirements of any and all research proposals that qualify or that have to use this standard, that it be proportionate and inclusive of underserved communities. And it's no longer just a friendly amendment, but that it is a part of the initial research proposal. And I don't know, as I said, if that's limited or it can apply across the board for everything from discovery to clinical trials.
Thank you, Ms. Duran. I second your wish, and perhaps this is something I'm sure that this is something the chair might potentially take forward in the future. But I think this is a good thing for the CERM team to work on, because I was somewhat struck when reviewers in this looking at this project, it was glaring, frankly, that the mismatch between the population that has been so disproportionately impacted in the United States by this epidemic and the HLA types that they were using in order to provide this vaccine. And I don't think that there's anything, any bad intent on researchers' part, but I do think it's important that we call this out and let the community know that this is important to us and a key value for CERM going forward.
This is
Oz. Could I make a comment?
Please.
So I totally support the sentiment and the motion in this case. The one problem is that we've always held to the concept that we should not rewrite the applicant's grants. And I think this borders on doing that a bit, and that it does impact a bit on review. So the more breaths you take, the less depth. And reviewers often criticize applications for trying to be too broad and therefore not being able to accomplish what they set out to do. I just raise all this now because going forward, I think it's going to be important to consider those points. Thank you.
Thank you, Dr. Stewart. Is there additional board comment?
Yes, Jeff, this is Steve Julesgaard.
Yes.
So could we hear the staff's thoughts about your proposed amendment?
Pardon me,
Well, we've heard from you about your proposed amendment and your concerns. I'd like to hear from somebody from staff.
Oh, from the staff. Sorry, I didn't hear that. Yeah, please. Dr. Sembrano?
Sure. So, I mean, those are amendments that we can certainly make. I think the only comment I would have is that for early stage discovery work, typically the work, is broadly applicable in most cases. But I think we can certainly make an amendment in an application like this in order to adjust it as requested. I think the applicant may be on the line as well and might be able to speak to the feasibility of doing so, but I think it is something that is certainly doable.
I do believe the applicant is on the line.
If the applicant would like to make a comment to that, I just again note that the reviewers.
Yes,
yeah,
hello.
please. Yeah, go ahead.
Oh, great. You can hear me. Hi, it is Albert Wong. I am the PI for that particular proposal. And first of all, we're working, as Dr. Sembrano mentioned, on a major HLA allele. Yes, it's present in the Western population, but that particular allele is also present in many of the populations affected by COVID-19. So it doesn't exclude them, that's for sure. Also, there is overlap between peptides that bind to HLA-A2. And I just called up the common HLA allele in African Americans as well as Hispanics and Asian population. So there is overlap between peptides that bind to HLA-A2 and HLA-30, 01, 29, O2, which is present in the Hispanic population. The first allele, 30.01, is present in the African American population. And then O2, O7 in the Asian population is actually extremely similar in its binding to HLA O2, O1. So I would expect a perfect concordance between the Asian population and the HLA A2 population. However, as was mentioned in the review, it is feasible to take those cells from affected populations from the Stanford Blood Bank and generate the stem cells as we contemplate in our proposal in order to create this autologous system and analyze it for binding. It is more work though. Certainly we can do it. Certainly we can accommodate it. I was going to speak during the public comment section. I mean, we could certainly accomplish this work. I think one reviewer did note in our review that, it's ambitious work and, that person did think that we could accomplish this. But I do note that the ICOC did approve for applications going forward a Discovery 2 budget of $250,000. Certainly, we could accommodate many analysis of many more alleles in our studies if we did have more supply money available for our research. So I'll stop and take your comments from there.
Thank you. So any additional board comments or questions?
Jeff, Isabel here. If nobody else has anything to say.
Yeah,
Please.
I'll go after you, Isabel.
Thank you, JT. I appreciate, I believe he said Dr. Wong. I appreciate what he said. What my concern is, and what I have seen in research grant after research grant, is there is more of an assumption that there will be inclusion as opposed to a reality. And too many times, particularly for communities of color, we see a promise of delivery, but we don't feel or see a result. And I think that for going forward, I would like to see that it is specifically addressed, that communities of color are included in research, and that there are ways in which this is actually shown at the end, on the other end, that in fact they were included, because I think this is important symbolically for many communities who have felt excluded from research and who oftentimes feel used but not served. And so while I think in some ways it could be just as symbolic, but I would love for it to be very clear, very plain, and puts us right there at I think the leading edge on calling on researchers to make this very clear and very plain that we are looking to serve the underserved and the under-researched. Obviously we don't, and I don't know all of the ins and outs of the science itself. So as you explain things, I'm going, oh, aha, well, that's good. But I do believe that we need to make it very clear and plain to the public what we are doing and that it's inclusive. And then maybe at some point in time, I can stop harping on this same issue. But right now, for me, it still feels like we're making assumptions that research will do what is right, as opposed to now requiring that they do what is right. And so I stand on the side of require to do what is right. Thank you.
Thank you, Ms. Duran. Chairman Thomas?
Yes, I would like to support the motion as well. I think that the comments that Ms. Duran has made, as well as others, we've now been addressing and commenting the need to adhere to these suggestions in general, but particularly here where you do have a disproportionate impact on certain communities, and you want to make sure to the full extent possible that work being done is going to address the needs of those communities. So I wholeheartedly agree. And I just, Gil, I wanted to ask you a question about, Dr. Wong has mentioned the budget issue here. And obviously we have no figures that address the increase of the budget to accommodate, get up to the 250 or whatever. But could you just respond to his comments, please, on that point?
So, everyone that is coming in at the moment is bound by the concept as it existed when they applied. So, meaning that the limit of their budget is 150,000, and so the activities that are proposed should fit within that budget. If there are additional experiments or activities, certainly applicants have to apply again and ask for additional funds. So a way of seeing the amount on a given project.
Well, perhaps Chairman Thomas could make an amendment to my amendment, increasing the budget, say up to another 100,000, since the applicant did mention the $250,000 limit, in order to do the work to make sure that the HLA is a representative of the communities most disproportionately impacted by the epidemic, by the pandemic, and that the decision on how much funds and how they are released would sit with the CERM team. So for instance, Dr. Sembrano, Dr. Wong would bring to you a suggested budget in order to cover all of the HLA types that are not covered, as he just described, going to the Stanford Blood Bank, and whatever that number is, maybe 5,000, 50,000, what have you, that the CERM team could negotiate that with them. It's like a fairly straightforward, simple thing to be done to make sure that we're actually developing a vaccine that will be available for communities that have been overwhelmingly disproportionately impacted by the epidemic.
So
this is Oz. Before there's a motion, could I make a comment about the general concept?
I think Chairman Thomas was coming in, and then I'll hear from you.
No, that's fine. I defer to Dr. Stewart. I'll speak after that.
Yeah, I wanted to actually say this before JT makes a motion, because I think it's an important point to consider. So we're kind of doing things on the fly here. And as everybody knows, I'm pretty much a procedure guy. And it worries me that we are trying to do things without actually having seen, for example, an application for how these funds would be used and having that reviewed by the grants working group. I'm sure any of the other applicants to this current round could have easily justified a budget of $250,000. I guarantee it. And they could have expanded it to make sure that they had sections on inclusivity. I think it's unfair to the other applicants to do this in an ad hoc way. As Gil said, it's possible for this applicant, Dr. Wong, to come back in at any time, request additional funds. It's very easy to do, and I'd rather do it systematically than ad hoc. Thank you.
Dr. Stewart, I take your point. I guess I'm sitting here in the Bay Area, and I'm struck that we've done community-wide testing in 2 different areas via UCSF. And in the overwhelmingly white, affluent community of Bolinas, we were unable to identify a single infection. Yet in an area of the Mission, predominantly Hispanic neighborhood, 95% of the infections were identified in Latinos. And these kinds of disparities exist up and down the state and across the country. And I agree that this is an unusual circumstance, but frankly, our responding to COVID is an unusual circumstance. And I think if we are intent on responding to COVID, I think we need to be, number one, exhibit the sense of urgency that communities and individuals who are confronted by this disease directly. And I think a little bit of flexibility in an agency that's spent over almost $3 billion at this point, over $50,000 to $100,000, seems rational in the face of a global pandemic.
I believe Dr. Martin also has a comment.
Sure, Dr. Martin.
I have a question. Jeff, your motion, the specifics of your motion, I would be interested in, because we heard from Dr. Wong that the HLA-A2 allele is clearly represented. I recall he said Hispanic population. The fact that it's high in the Western European is not so relevant as to me as whether the underprivileged, highly vulnerable population is not adequately represented by the HLA-2 haplotype. And so I wonder whether it could be, I don't remember exact words of your amendment, but could it be reworded such that they were not disadvantaged by the primary focus of this research grant in terms of the prevalence of the HLA-A2 allele?
I suppose, and I just would like to remark that I'm operating off concerns that were expressed by several reviewers in the GWG. This is not a field where I obviously have the depth of knowledge to be able to describe what it takes to make sure that this project would cover populations disproportionately impacted by COVID. But and perhaps Dr. Wong, would that help Dr. Martin to hear from Dr. Wong again, if he's still with us on?
Yes, it would, yes, it certainly
Dr.
would.
Wong?
That would help. I mean,
Yeah,
Yes,
thank you.
hello, hi, I'm on the line. Go ahead.
Please. Dr. Wong.
so
Yes.
So this is Dave. My question simply, Dr. Wong, is the HLA-A2 haplotype underrepresented in the populations, and particularly California, that are disproportionately infected by COVID-2?
Well, actually, first of all, HLA allele types affected by COVID-19 is actually one of my pet interests because I am quite aware of how different populations have been affected. So this is why I have this information up right in front of me. So as far as HLA-A201, it is in the African American population, it is the 10th most common. Actually, what I've got is the dual allele type. But for the O201 allele, which is what we're looking at primarily for this application, it is highly represented. I want to say approximately 30% of African Americans would have it. It is the 10th most represented allele when you consider it with its HLA-B subtype. In the Asian American population, it is the second most common allele type. And in the Hispanic population, it is again, it's the 9th most common allele type. So there is representation there of those populations that are disproportionately affected by COVID-19. And also, I do expect that peptides that bind to HLA-A2 will also bind to the common alleles found in these other populations because there has to be overlap, right? We do react very similarly to antigens, and so I would not be surprised if once we delved into it and looked at it, that we would find some commonalities. But certainly addressing that population directly would be the most efficient. I understand the concerns that there's been no formal vetting of any budget, but certainly we could submit a budget proposal related to how we could expand our work, and I would be more than happy to look at it. It is, as I said, I do believe that HLA subtype is one of the underlying predispositions to COVID-19, and I would be more than happy to investigate that in my lab.
So this is Oz, and again, could I respond to Dr. Wong's comment and suggestion there, because I think there may be sort of a middle ground here. Again, what I was objecting to was sort of an ad hoc addition of $100,000 to a budget that had been reviewed by the grants working group. If we were to consider a motion, I would be much more favorable. I'm not sure I'd vote for it yet, but I'd at least be more favorable if the proposal was to submit an amendment for additional budget that would be reviewed by the grants working group. The GWG is meeting quite often, regularly, and so having this consideration could be done in a matter of weeks, and then could come back to the board for formal approval as such. Thank you.
That a motion?
So, Mr. Sheehy,
Yes, Chairman Thomas.
I'm very mindful of Oz's procedural points. I am, however, very mindful that in making this award, we want to make sure that as it's defined at the moment, it meets the goals of, as discussed by Ms. Daron and others. And if we approve this, and it goes, and we have, say, an amendment of the sort that Oz just proposed, and it goes to the GWG, and for whatever reason it does not pass there, then if we vote on this project today, which I am in favor of, we may find ourselves in a situation where we haven't adequately covered the scope of what we wanted done in the award as we're voting today. So I'm a little, I think, Oz, are you suggesting that we approve this today and then take an amendment to the GWG, or are you proposing that we don't approve this pending a review of an amendment?
No, I'm proposing that we approve it, that Dr. Wong be asked to submit what NIH would call a supplemental application. It is already a little bit out of our normal mode of operation, but I think it's something that is at least in keeping with our general concept of the procedural reviews. That could be reviewed by the GWG and come back to us at the next ARS subcommittee meeting. Thank you.
Okay, thank you. So, and I believe that, Gil, the next GWG is a week from Tuesday, is that correct?
Yes, that's correct.
Okay, so I then would move that we do approve this today, subject to Oz's amendment to have Dr. Wong submit an amendment to the budget to the GWG for review at the next meeting.
So Chairman Thomas, we do have a prior motion. As the maker of the motion, I would be willing, I think Ms. Daron was the second to that motion. So the question is whether we would be willing to accept that as an amendment to our motion, I believe is probably an appropriate way to go. Ms. Daron?
Mr. Sheehy, first of all, I appreciate procedure and process, et cetera. So I'm perfectly open to an amendment. I'm hoping that in the future, all of these kinds of amendments can be avoided if the call for proposal is very clear at each stage and has some kind of requirement of the researchers to present, their plans for inclusion, et cetera. But in the meantime, I will go along with how you restate the proposal, the amendment.
Okay, so what I'm proposing then is that we accept Chairman Thomas's amendment to our motion to approve this. And so the motion would be to approve 11941 with a condition that that Dr. Wong bring to expeditiously to the grants working group to CERM a request for supplemental funding in order to complete the HLA work necessary to make sure that we capture that we are inclusive in the development of this product to every to the communities that have been disproportionately impacted by COVID. I think that's agreeable to everyone. And
So what happens to the first two applications? Are they included in your motion? So we handle it all in one motion?
if you would like to do that, we can include those two as well, Senator Torres. I'm happy to do that.
Great.
Is there any objection from any of the other board members? From Ms. Doron, do you mind if we include the other two? Go ahead and fund the ones that are fundable, since I think you are the second to the original motion.
No, I do not mind. Yes, I'll.
Okay.
That's
So
not.
with that, is there any board discussion to the approval of all three applications in tier 1? Is there any public comment? Ms. Bonneville, could you call the roll?
Anne-Marie Dulege,
Yes, I approve.
Isabel Duron,
Yes.
David Higgins,
Yes.
Steve Julesgaard,
Yes.
Dave Martin,
Yes.
Lauren Miller, Adriana Padilla. Adriana, are you on the line and I just can't hear you, or did you drop off? Let me look.
Can you hear me?
Oh, now I can.
Yes.
Thank you. Joe Panetta,
Yes.
Francisco Prieto,
Aye.
Robert Quint, Al Rowlett,
Yes, and thank you for the discussion.
Jeff Sheehy,
Yes.
Oz Stewart,
Yes.
Jonathan Thomas,
Yes.
Art Torres,
Aye.
Diane Winokur. Let me see. Diane is on mute, but I'm going to see if I can. Diane, you're on mute. Can you unmute yourself? Sorry. Yes. Okay. I see the thumb. Thank you. The motion carries.
Great. We have one last motion, and that motion would be not to fund any of the applications in tier 2. Do I have a second?
Second.
Seconded by Dr. Prieto. Any discussion by the board? Any public comment?
I believe there was public comment. Doug, did you see a hand? I thought I saw a hand raised.
Evan Snyder, please.
Okay.
Okay. Dr. Snyder?
Is it my turn? Are you ready for me to go?
Yes. And 3 minutes, please.
Okay, I certainly will do that. I think, as you can see, we are right on the borderline. And apropos to the conversation we just had, I think this is extremely pertinent. So a quick way of background, our institute, thanks to Sumit Chanda, did an unbiased high throughput screen of 12,000 FDA approved and clinical stage drugs, and came down to a list of 30, and from that list to 6 that clearly inhibit the cytopathic effects of SARS-CoV-2. However, as you know, all of these high put through screens are done on clinically irrelevant cells. What we have is something called a lung organoid system.
Excuse me, Dr. Snyder, we don't know what item you're referring to. What is the number of the grant?
I'm sorry, it's it's the second one. It's
11782.
Thank you. Okay, and it's a very long title, but pretty self-explanatory. Anyway, as you know, there is no good animal model. However, most of these drugs have already not only been in animals, but already in humans. There is in many of them, a complete IND package in the FDA. What is needed is simply a clinically relevant system to help prioritize those drugs to fast track to clinical trials. And IQVIA is helping with this. This lung organoid system is acknowledged by the study section as being novel and powerful. It has all of the cell types, both proximal and distal. It's invested with macrophages, which mediate inflammatory reaction and cytokine storm, and the vasculature, all IPS-derived isogenic, which can also talk to the pulmonary edema aspect. Importantly, because these are IPS-derived, it addresses the concern that one had. These are patient-specific lung organoids. In the pipeline, we are making lung organoids from a variety of races, including African Americans and Caucasians, from male versus female, from a variety, particularly the HLA mutation that has been documented to be most susceptible to SARS-2 infection. It provides a developmental profile so we can figure out why is it that kids may be less vulnerable, but when do they become vulnerable? These are the only lung organoids of this kind in the world's literature. But as you can imagine, if it comes out of this, California would be the leaders in the only authentic system providing a tool for screening, not only these drugs, but other future kinds of therapies. In addition to some more esoteric answers like, what is the mechanism of action of the virus? What cells does it really bind to? What's the mechanism of action of the drug? We were criticized for only two aspects. One, the bandwidth. At the time, we only had 3 people working in the lab. Now we have 12, mostly from volunteers who are so enthusiastic about doing this that they want to be part of this effort. And then preliminary data, not on the integrity and the value of the system, but specifically with SARS-2 infection. And they were asking for any viral data for any virus. Well, in fact, not only do we have some promising data with the pseudovirus, but the lung organoid system was developed initially for being gene modified with a lentivirus, lentiviral mediated gene transfer, which of course shows that it is infectable with a virus.
Dr. Snyder, your 3 minutes have passed.
Can I say one more thing, or is that?
Quickly, please. Okay.
I just wanted to make one comment from one of the reviewers that said, given that they, meaning our group, are an experienced lab with good expertise, and there was enthusiasm about many aspects of the award, I lean towards giving them an opportunity. And I would think that in a place where it's unfunded, just too is the perfect mechanism for allowing us to do this.
Thank you, Dr. Snyder. Is there additional public comment to this motion? Ms. Bonneville, could you call the roll?
Yes. Anne-Marie Dulege?
Yes.
I'm sorry, are we voting to whether or not to move this forward? I'm not sure what we're voting.
I'm sorry, Mr. We are voting to not fund any of the remaining applications, which are the ones that were in the unfundable scoring range. So these were the ones that were not recommended for funding by the grants working group. And so in order
Mr. Chair,
we need to specifically vote not to fund them.
Dr. Prieto, did you have a question as well?
yeah, no, I think I just clarify my understanding would be that if we wanted to move another application back into the fundable range, you would have to vote no on the current motion, and then we would need a substitute motion to move that application up, vote on that motion, and then return to this motion. So if you want to not move anything and proceed as we were originally going, then you would have to vote yes on the current motion.
That's correct. And I just would note that we're reviewing constantly now. So applications can be resubmitted and re-reviewed as we just did with Dr. Wong's application in a fairly expeditious period of time. So concerns could be addressed in that period. But anyway, yes, so that's the motion. So Ms. Bonneville?
Sure. Anne-Marie Dulege,
Yes.
Isabel Duron,
Yes.
David Higgins,
Yes.
Steve Julesgard,
Yes.
Dave Martin,
Yes.
Lauren Miller, Adriana Padilla,
Yes.
Joe Panetta,
Yes.
Francisco Prieto,
Aye.
Robert Quint, Al Rowlett,
Yes.
Jeff Sheehy,
Yes.
Oz Stewart,
Hold on, please.
Yes.
Jonathan Thomas,
Yes.
Art Torres,
Aye.
Diane Winiker. Let me see, Diane, is Diane giving me a thumbs up? Diane? Diane?
You're on mute, Diane, so you have to give.
I am muted.
Oh, there you are. Is it, I didn't hear, I couldn't record your vote.
Yes.
Thank you so much. The motion carries.
Thank you. Chairman Thomas, that concludes the business of the Application Review Subcommittee.
Thank you, Mr. Sheehy. Thank you for all participants in that discussion. We now, I believe, have approved our 7th, 8th, and 9th COVID-related project, and we will undoubtedly have more interesting ones to come in the future. So thank you. We are at the stage of the agenda for general public comment on any items whatsoever. Do we have any public comment?
We do not.
Hearing none, that concludes the.
JT, can I make a comment, please?
Certainly.
Thank you. I guess in listening to Dr. Snyder, my head moves, my heart moves towards, thinking about what he said as opposed to my head knowing what the heck they're talking about. But I was looking at the no votes, and there were like 11 no votes on this project. And so part of me wants to support him coming back, but with 11 no votes, I don't know what chance he has. So I'm just saying that I get torn between the emotional aspect of wanting to support something and give it a try versus the science says it's not ready for prime time. So I appreciate all of your guidance on this. It just once in a while, I like to take a flyer on this, let's do this, but I will follow your guidance on that.
So I just would like to concur with Mr. Sheehy's comments on this, and thank you for that, Ms. Daron. This is a matter of, according to Dr. Snyder, a couple of critiques that were specifically made, which he has addressed, and in doing that, he's welcome to resubmit and have those critiques reevaluated. We have that process done all the time. And this is a very short-term road to reconsideration. So I do want to recommend to Dr. Snyder and any others who have applied, they're more than welcome to reapply and address the concerns referenced in the comments. Okay, so hearing no other comment by members of the board or the public. Thank you, everybody. Please continue to stay safe and healthy, and we will be reconvening shortly for the next round of discussion on these grants. Thanks very much.
Both, don't forget. Bye.
Bye-bye. Thank you.